I had the pleasure of introducing Dr. Daniel Perez. Dr. Perez is the Caswell S. Edison Chair in the College of Veterinary Medicine, University of Georgia. Dr. Perez has approximately 30 years of experience working with influenza viruses. His laboratory has developed a monoclonal antibody currently used for rapid diagnosis of the avian flu. Dr. Perez's laboratory is one of the centers of excellence of influenza research and surveillance established by the NIH. More recently, in response to the SARS-CoV-2 pandemic, his research group has embarked on in vitro and animal studies to better understand the pathogenesis of this virus, along with the testing of antivirals and new vaccine strategies. At this time, I'll turn the floor over to Dr. Perez, who will present on One Health and the potential of H5N1 jumping from birds to humans. Dr. Perez, you have the floor. Thank you.
Thank you, Kevin, for the invitation to talk today. Today, I will be telling you a little bit about H5N1, the chances of transmitting from birds to humans, and how concerned we should be about avian influenza in general as viruses of pandemic concern. So for those of you who are not familiar with influenza viruses, they belong to the Orthomyxoviridae family. The most important genus within that family are the alpha influenza viruses or type A influenza A viruses. What these viruses have in common is a lipid bilayer derived from the host plasma membrane. These are pleomorphic particles with a diameter that ranges between 80 to 120 nanometers. You can have, the virus can show itself on the electron microscope as semi-spherical particles, bullet-shaped particles, elongated particles. And those that circulate in humans tend to have this elongated shape, which is thought to favor transmission. The genome of these viruses is an RNA genome that is segmented of negative sense, and the virus has an entire cycle based on RNA, and for that, it has an RNA-dependent RNA polymerase complex. So influenza viruses are, because they are RNA viruses, and because the polymerase complex does not carry proof reading activity, they're constantly changing. The virus tolerates mutations, particularly on the surface. There are two major surface proteins, the hemagglutinin and neuraminidase, or HA and NA. When these mutations accumulate on these proteins, it causes antigenic drift, and that leads to prior immunity not being able to stop the drifted variant. In addition, the virus, because it is segmented, it can undergo reassortment. So that's an exchange of gene segments. And when that exchange of gene segments involve the hemagglutinin, that can lead to antigenic shift, which is what happens during pandemics, the replacement of an old HA circulating in the population for a new one, or a virus that carries a new HA protein that is antigenically very different from what was circulating previously. The virus replicates in the nucleus. I just put a very simple cartoon of the replication cycle. The virus recognizes receptors. These are sialic acids on the surface of the cell, enters the cells by endocytosis. Within the endosome, the acidic pH of the endosome triggers the activity of M2. The M2 is a proton pump that is essentially a unidirectional proton pump that brings protons into the interior of the virus that causes changes in the hemagglutinin. The hemagglutinin exposes a fusion peptide and allows the effusion of the viral membrane with the endosomal membrane and the release of these H segments in the form of viral ribonucleoprotein particles towards the nucleus of the cell. The virus uses a particular mechanism of synthesis of messenger RNAs, of viral messenger RNAs, by stealing short cap structures from host cellular messenger RNAs and primes for the synthesis of its own messages. Some of the early proteins to be synthesized are the non-structural protein NS1 that has essentially an anti-interferon activity, so it has an antiviral activity. Later in the replication cycle, once progeny ribonucleoprotein particles have been produced, the assembly occurs at the level of the plasma membrane, where all these H segments, through specific signals, recognize each other and recognize the matrix and find the HA and NA on the surface of these bad virions, and the virus gets released. So there are antivirals known as M2 inhibitors, polymerase inhibitor, or the PI subunit inhibitor, and the neuraminidase inhibitors. These are essentially the three large classes of FDA-approved drugs that we have, or for which we have drugs approved. But unfortunately, influenza viruses tend to make resistant to any one of these inhibitors fairly quickly, and usually these viruses remain infectious and transmissible. Over the past 20 plus years, we've been lucky to have a system called reverse genetics, essentially cloning all 8 segments of the virus in plasmids and transfecting those plasmids into cells and starting a replication of influenza virus de novo. And that has allowed us to study how the viruses causes virulence, which are the roles of every protein in the virion, finding that there are additional protein or small protein products encoded in the virion, and also to improve methods for vaccine development and antiviral screening. The natural reservoir of influenza viruses are wild aquatic birds of the world. And in those birds, we find influenza viruses divided into subtypes based on the antigenic differences of the HA and NA. So there are so far described 16 HA subtypes and 9 NA subtypes. Only the subtypes known as H5 and H7 have the potential to cause outbreaks of high morbidity and mortality in poultry. FLUAVS A influenza viruses have been isolated from 90 species from 12 orders, mostly from Seriformis and Caradriformis, and through their migration routes, play a big role, a key role in maintaining influenza viruses and disseminating these viruses. The replication in these birds is usually a gastrointestinal replication or an intestinal tract. The transmission is fecal oral through bodies of water. And that's how the virus is able to perpetuate itself in nature. In these species, usually it's thought that the virus causes no disease, although it is not really possible to establish whether it causes disease, although we don't see overt clinical signs of disease in birds affected by influenza. In addition, more recently, subtypes H17 and 10 and H18 and 11, these are flu type A-like influenza viruses. They're not quite the same as those circulating birds in terms of the structure of some of these proteins. And they have been found in fruit bats in Peru and Guatemala. And more recently, viruses called H9N2-like have been identified in fruit bats in Egypt. So flu viruses are known to be able to jump species. So from this large reservoir, we have all these 16 HA and 9 NA subtypes. And occasionally these viruses can transmit to other species. And those include sea mammals, horses, mink, occasionally pigs, and land-based poultry. I have to emphasize that land-based poultry are not considered a natural host of influenza. Usually when a virus from a wild bird jumps into a land-based bird, it is expected to see changes in the virus before it can establish itself in a new species. An example of viruses that have become essentially adapted to poultry are the highly pathogenic H5 viruses, low pathogenic H9 and 2 avian influenza viruses, and others. And from these land-based poultry, occasionally you may have viruses that can jump to other species like pigs or humans. In pigs, we have the notion that they are considered mixing vessels of influenza because they are susceptible to both human and avian influenza viruses. And considering that the influenza virus genome is segmented, there is an environment in the pig that can favor gene segment exchange and generate novel strains. And that's what we thought that has happened, at least in some of the pandemic influenza strains that are emerging and in humans. One particular aspect that restricts the host range of influenza viruses is the recognition of receptors. Receptors for influenza are sialic acids found on glycoproteins of the cell. And these sialic acids can be presented, depending on the species or the tissues, in largely two forms: linked to the adjacent galactose residue in an alpha-2-3 linkage or in an alpha-2-6 linkage. So when viruses are able to jump to other species, if those species carry in some of those tissues or tissues that are susceptible to flu replication to 6 receptors, then there is a chance that with time, these viruses may evolve the ability to bind TU6 receptors, which are also found in the respiratory tract of humans. And that is the case of pigs and some species of land-based poultry, in which the respiratory tract has a combination of both TU6 and TU3 receptors in different proportions. To highlight a little bit further the importance of receptor specificity, I think that those viruses that affect humans known as H3N2s, it is very clear that a change or two changes in amino acids from viruses that recognize alpha-2,3 linked sialic acids that contain a position 226 glutamine and a position 228, when the virus mutated with a leucine at position 226 and a serine at position 228. That was enough to confer the virus ability to bind to 6 receptors. These are very small changes, and that can have a big impact on the host range of the viruses. And in the case of the H3N2 pandemic strain of 1968, this is one of the features that probably allow the virus to cause a pandemic in humans. But the ability to change to a TU6 receptor binding virus preferentially is not restricted to a virus that replicates in humans or in pigs. It can also happen in land-based poultry. This is the case of the H9N2 low pathogenic avian influenza viruses, which are endemic in many parts of the world, particularly in the Middle East, Africa, and all over Asia. These viruses have evolved with a mutation, position 216, which is the equivalent of 226 in the H3HA. Most of these viruses carry leucine at this position. The second most prevalent population of these viruses carry glutamine in that position. And the difference of this single amino acid makes a big difference in the virus ability to recognize TU6 receptors. What you're seeing in the graph there are the representation of binding ability to array in a glass that has glycans, different glycans that contain either on the tip of the molecule a 2,3 receptor or a 2,6 receptor. If you look at the wild type H9N2 with leucine 216, you will see that even though there is a proportion of receptors, both 2,3 and 2,6 that are being recognized, the highest signal comes on the 2,6 receptor. If you mutate this virus and incorporate a glutamine 216 instead of leucine 216, the virus now becomes mostly a 2,3 binding virus and loses the ability to recognize 2,6 receptors. If you do the opposite, if you take a virus that has a glutamine at position 216 and move it and mutate with a leucine at 216, now the transition from 2,3 preferential binding goes to 2,6 preferential binding. So that is to highlight that it's not only in humans that you can have viruses, what is called human-like receptor specificity. There are species of poultry, chickens in particular, that carry both types of receptors. And in the case of the H9N2s, they have evolved the ability to bind to 6 receptors. If you ask me, of all the viruses that are circulating out there, the one that has the most features that resemble a human influenza virus are in fact H9N2s and not H5N1s. So from one of these reservoirs, whether it would be poultry, ducks, or pigs, you can have pandemic influenza strains emerging. That was the case of 1918, H1N1, then again in the H2N2 in 1957, and then again in 1968, H3N2. Every time we have those pandemics, the previous subtype that was circulating in humans was eradicated. So in 1957, there was a natural eradication of H1N1. It was replaced by H2N2. In 1968, there was a natural eradication of H2N2, and it was replaced by H3N2. The exception is the reemergence of the H1N1 in 1977, known as the Russian flu strain, probably released in Asia in some place, not necessarily Russia. But the difference here is that despite the fact that this virus basically was spread and incorporated back into the influenza viruses that were seasonally present in humans, that virus was not enough to replace the H3N2. This is all along with the presence of influenza B viruses that in the late 70s evolved into two major lineages known as Yamagata-like and Victoria-like. And as you probably know, the emergence of the SARS-CoV-2 virus the measures that were taken to control the infection, the use of masks, social distance, etc., may have led to the extinction of the Yamagata-like viruses. They haven't been seen since prior to the 2020 pandemic. Now, prior to the SARS-CoV-2, we have the pandemic of 2009. That was an H5N1. So how can it be that the H5N1 pandemic strain caused a pandemic if we had a seasonal H5N1 circulating in humans? That is because that H5N1 virus was antigenically so distant from the previously circulating H5N1. And in addition, it was a virus that replicated efficiently in humans and transmitted efficiently in humans that led to the extinction of the H5N1 seasonal strain. So since 2009, there is no more 1977 descendants of the H5N1. We only have 2009 pandemic strains co-circulating with H3N2s, and now with the Victoria-like lineage of Influenza B viruses. So the evolutionary history of these viruses is very complex. Every time there has been a pandemic, there has been an avian reservoir that donated some of these gene segments. In the case of the H5N1, this may have been an entirely avian virus that went into both pigs and humans and caused the 1918 pandemic. In 1957, the pandemic strain acquired 3 gene segments from the avian flu reservoir. Those segments were the HA, the NA, and the PV1 segment. And then in 1968, the virus inherited 2 gene segments from the avian reservoir, the HA, H3 subtype, and the PV1 segment. The pandemic H5N1, although it was of pig origin or a virus that was circulating in pigs, there is a very complex history of that virus that has gene segments derived from the avian reservoir, the human influenza viruses, and pig influenza viruses, and from different continents, from North America and from Asia. And I anticipate the next influenza virus is also going to have very complex evolutionary histories with the participation of viruses from many parts of the world and not just from a single region, if we let them be the way we are right now. So avian influenza itself is a disease in birds, in chickens particularly, and other land-based poultry. It was originally described as fowl plague in 1878 by Peroncito in Italy. Avian influenza can cause disease outbreaks of varying intensity in poultry, and they are essentially classified based on pathogenicity in chickens as low pathogenic avian influenza viruses or highly pathogenic avian influenza viruses. Most influenza viruses are low pathogenic influenza viruses. And outbreaks with these viruses don't cause overt clinical signs, but they do affect growth in meat chickens and drops in egg production in layers. The highly pathogenic avian influenza viruses are, as I said before, restricted to the H5 and H7 subtypes, and they become so by accumulating a polybasic cleavage site on the hemagglutinin, on one of the surface proteins, as I will explain later why this is relevant. So highly pathogenic influenza viruses contain this polybasic cleavage site, but if they don't contain a cleavage site, in times when we did not have sequencing tools to establish whether they had or they had not a polybasic cleavage site, most of the establishment of the classification into high path versus low path was based on index tests called intravenous pathogenicity index. This is essentially growing the virus in eggs, making 1:10 dilution and inoculating 10 chickens intravenously with that dilution and waiting for mortality to happen over a period of 10 days. If that index test is above or equal 1.2, the virus then is classified as high path. So although a polybasic cleavage site is likely a condition for an H5 and H7 to become highly pathogenic, there is also the possibility that other viruses, even other subtypes, could become highly pathogenic, and they will not show a polybasic cleavage site, but they will show a high mortality in the intravenous pathogenicity index test. In the United States, these highly pathogenic influenza viruses are considered select agents, and there are obviously rules and regulations and biosafety precautions that we have to abide to in order to work with them. Now, the cleavage site of the hemagglutinin is important because that leads to infectious virulence. So the hemagglutinin of HA on the surface of the virion is present as a homotrimer. So 3 monomers make essentially one HA unit. And there are hundreds of these trimers on the surface of the cell of the virus. Now, when the protein is synthesized in each monomer in a metastable state. And that metastable state has to be, its cleave is the subject of trypsin-like proteases in the extracellular compartments, normally for low pathogenic influenza viruses and for human influenza viruses as well. And that cleavage, then after it occurs, those 2 subunits that are generated are linked by disulfide bonds. And that new structure that is set up is the one that becomes infectious, and the one that once it reaches the cell and in the endosome, acidification leads to the expression of a fusion peptide and the fusion between the viral and endosomal membranes. So in the case of low-path avian influenza viruses, these cleavages occurs, as I said, by trypsin-like proteases present in the lumen of the respiratory and intestinal tracts. But in the case of the highly pathogenic avian influenza viruses, the HA is cleaved by subticillin or furin-like proteases found associated to the ER. So that leads to the HH to change or to mature within the cell compartment and allow cell-to-cell spread. And that is the reason why in poultry, highly pathogenic influenza viruses are able to cause systemic infection. You may have seen pictures like this before, but you can read the many features and clinical signs that may be associated with a highly pathogenic avian influenza outbreak. But none of these descriptions are unique to a highly pathogenic avian influenza virus. There are other diseases that affect poultry. For example, virulent Newcastle disease virus, or here is called exotic Newcastle disease virus, that can have similar effects on the bird. And sometimes there are no effects on the bird. You see a poultry production in which chickens look pretty normal, and in 2 days they are all dead. And there was no warning signs about any infection in your flock. So how bird-to-bird transmission occurs? Well, through oral or nasal routes and in the feces. Fecal to oral transmission is the most common, but humans can also transmit bird flu, particularly long distances, okay? So that's why biosecurity is so important to prevent avian influenza. And there are certainly other factors that may lead to transmission of avian influenza, but I would say that outside from bird-to-bird contact, the second most common contributor to the spread of bird flu are humans through their activities, not that they get infected. So one of the things that we have done so well, and this is not a farming practice that is new, this is actually very, very old, thousands and thousands of years old, this integrated fish farming, which is very, very common in Asia, is probably one of the most environmentally friendly systems for sustainable agriculture, okay? But it can also promote the emergence of influenza A viruses. And you can see why, because we have here a fish pond in an animal production system. This could be a pig production facility, could be a duck production facility, could be a chicken production facility, in which nothing goes to waste. The waste generated by each animal species is used to essentially feed the other animal species. Essentially, that's how more or less it works. But in a place like this, you create the conditions for influenza A viruses to really flourish and to create the conditions for interspecies transmission. Another system that perpetuates and perhaps amplifies influenza A viruses are live poultry markets around the world. And you know that with the emergence of the H5s and H7s and many other influenza viruses that have affected humans, recently H10s, they all have basically a source in live poultry markets in Asia. One of the problems with live poultry markets in Asia is that they are the breeding ground for these, what I told you before, H9 and 2s. These are the most prominent live low pathogenic avian influenza viruses in the world. They are in many commercial and backyard operations and poultry markets in Asia, the Middle East, and Africa. And they were implicated in the genesis of both the H5N1 highly pathogenic influenza viruses in China and in the genesis of the H7N9 low path and then the transition high path avian influenza viruses in China that have infected humans. Unfortunately for us, H9N2's low pathogenic avian influenza viruses are not a notifiable disease. Countries are not obligated to report H9N2s, and therefore they circulate freely, and sometimes where we know of their existence is years after the fact. That complicates our ability to control other influenza viruses like the H5s and H7. So I believe we have to do a better work of controlling low path avian influenza viruses in general, particularly in poultry markets. I always say, and this may be like a joke, but there is some truth to it, that H9N2 low pathogenic influenza viruses are sort of like the godfather. They don't go and do the dirty work. They send other viruses to do the dirty work, and those are the ones that have been on the news infecting people. But I think they are a reason for concern. So that brings us to the emergence and evolution of the H5N1 bird flu, started in, or at least detected in 96, 97 in geese, but when it was discovered already, had all the hallmarks of a highly pathogenic avian influenza viruses. 18 people were infected, 6 of them died, and essentially culling of the and closing of the live bird markets in Hong Kong back then when this occurred led to essentially stopping all the human cases. But the virus then reemerged, actually never went away in mainland China. So there was an explosion in 2003, 2005, with the virus spreading beyond the borders of China and further evolving into multiple clades or genotypes. Then in 2014, 2016, these H5s show little change by exchanging the N1 and N subtypes for those of N6 and NA subtypes. Those were also found in gene segments usually found in wild birds, most likely from a source in a live poultry market. And that led to the export of these viruses beyond poultry into wild birds and traveling through migratory routes into other parts of the world in Asia, Africa, Europe, the Middle East, and the virus reached North America in 2014, where it caused in 2015 outbreaks in both turkeys and chicken layer operations in the Middle East. In 2018 through 2020, we see that these H5N6, H5N8 viruses become predominant globally, but they reassert once again. They inherit an N1 from a wild bird virus, and the virus continues to be spread. And it gets to the 2021, 2022, where we have this unprecedented spread of the highly pathogenic avian influenza virus, H5N1, and clear evidence that there is a two-way transmission between poultry and wild birds. Viruses that went from poultry to wild birds and then from wild birds to poultry again. So the evolutionary history is complex, but essentially through mutation and reassortment, the virus has diversified and led to what we have today, in which basically, except the Antarctic, the rest of the world has been dealing with, obviously not every country, but most continents have outbreaks of these viruses, including Central and South America. And this is the first time that you have outbreaks of Eurasian lineage goose 1 dung H5N1 highly pathogenic avian influenza virus in both Central and South America, both in poultry and in wildlife. And when you look at analyzing the HA sequences of these viruses are very similar. They are all derived from this introduction into early in North America. So in the US, we have had outbreaks in commercial poultry, backyard poultry, wild birds, and wild mammals. In the continental US, I don't think there is a single state that has not reported a wild bird isolate of H5N1. So it's widespread. And in many cases, it's associated with high mortality. It has also affected the commercial sector. Basically, close to 60 million birds have been either died or culled because of these outbreaks. The most affected states are Iowa, which is a place where you have basically layer production. So there is a large number of egg-producing chickens in there. And again, most states have reported outbreak except for Louisiana and West Virginia. Every other state has essentially detected the virus in their either backyard or commercial flocks. And then we get to perhaps most concerning part, the ability of the virus to infect mammals. The ability to infect mammals has not been just restricted to wild mammals in the US, but other parts of the world. The most perhaps important outbreaks have been in sea lions in Peru and in a mink farm in Spain. I know you probably have seen in the news some of the concerning features of these viruses, the identification of what are being called mammalian adapted mutations, particularly in the PV2 of the virus identified in minks in Spain. There is a mutation that improves polymerase activity. But I have to say that, to me, we are probably jumping into conclusions about the ability of these viruses to become more adapted to mammals. I don't think these viruses are any more adapted to mammals than what they were in 2003, 2005. What is really concerning is the geographic spread, because by having expanded geographic spread, then you have more opportunities of infection of species in which we do not know what the behavior of the virus is going to be, and which of those species may be able to generate a strain with unusual phenotype that can make it more transmissible to humans. But so far, the evidence does not suggest that these viruses are any more adapted to infect mammals than when they were in 2003, 2005 at the peak of poultry outbreaks back then. Therefore, I would use caution when reading information that says that some of these viruses may be gaining the ability to infect mammals more efficiently. I think this is, at this stage, an effect of opportunity. So these animals feed from either birds that are too sick, that are infected and are too sick and cannot fly, or carcasses, and therefore probably they die from highly pathogenic influenza. They have high virus loads, and that's what is infecting these animals. And this is not different than what infected humans before, in which the notion that we have is that, yes, humans can be infected with these viruses, but to infect a human, usually you need to be in an environment or in contact with an animal, or again, an environment that has a very high load of virus in order to infect a human. I don't know if that is clear, but I wanted to make it as clear as possible. To me, the most concerning part is the number of opportunities that we have to get exposed to low pathogenic avian influenza viruses in places where we know they do circulate, like poultry markets in the world. And yes, H5N1 can have devastating infection in a human, but again, you may need very high doses of virus to infect. I have to remind always everyone that when we talk about pandemic influenza, the H1s, the H2s, the H3 subtypes, they cause almost no disease in poultry. So you don't have to have disease in poultry to have a pandemic in humans. In pigs, H1 and H3 infections, they do cause respiratory infections, but the most significant effect of this flu infection in pigs are when they are associated with other factors, other infections that are part of the porcine respiratory complex. Per se, influenza viruses in pigs cause very little disease. Again, I'm not downplaying the effect that flu has in pigs or in poultry, but I just want to say that when we think about pandemic influenza, we should not think about the highly pathogenic viruses in poultry basically means a more or a nastier pandemic in humans. It doesn't necessarily go together. But they do cause infection in humans. Since 1997 to 2023, we have had 869 cases of H5N1 infections in humans and almost 460 deaths. So the virus can be really nasty. But if we look at what happens with these newly emerging viruses from 2020 to 2023, the number of human cases have been very, very few. So to me, there is no any clear indication that these viruses are any worse or any better at infecting humans than what they were back in 2015, 2003, etc. However, when we look at H9N2 cases, they seem to be on the rise in the last few years. These are low pathogenic avian influenza viruses. They cause mild disease. There have been only 2 deaths associated with these infections in 2 patients that have other comorbidities. So they were basically at a disadvantage when they had this infection. But if you look at data on seroprevalence of the infections, maybe the exposure that we have with these viruses is far higher than what we have with other viruses that we are concerned about, like the high path H5N1s. So that's something that I would like to bring to the table because it's often ignored in the media. The importance that these H9N2 viruses may have in the overall ecology and evolution of strains of influenza with pandemic concern, whether the next pandemic is going to be caused by an H9N2 virus or any other subtype, these viruses are able to infect humans by themselves and have participated with donating gene segments to viruses that have zoonotic potential. And certainly, the goose, guangdong, H5N1 are a threat to food security. And in my modest opinion, we do have a pandemic of H5N1 in wildlife. So I think that that's something that we need to be concerned about. We don't know how this virus is going to affect wildlife biodiversity and ecology when we have these instances of mass mortality in some species, some of them which can be endangered. Certainly has had a big impact in poultry worldwide, not just in the US, but all over the world. And I think at the beginning, Kevin was talking about inflation, higher interest rates, and the prices of eggs or something like that. But the H5N1 outbreaks are certainly not helping with poultry production around the world. They are becoming a problem. So what options do we have in poultry? Most countries rely on culling of infected birds or infected premises because of trade restrictions. H5s and H7 highly pathogenic viruses are notifiable disease viruses. So vaccination is not usually seen in countries that have a big component of poultry production into exports. They don't consider vaccination. So they handle most of their outbreaks by biosecurity or culling of infected premises. But other countries that are more importers than exporters of poultry have relied on vaccination. In the US, there is no vaccine approved or in use in commercial poultry against highly pathogenic avian influenza viruses. Again, because vaccination negatively affects poultry exports. 99% of the vaccine that has been used has been in Mexico to control H5N2 and H7N3. There is no indication that these viruses from Mexico are able to infect humans easily. And vaccination has been used, I would say, successfully to control clinical signs, to control the overt clinical signs in poultry production. But they have not really eradicated the virus from their production system. China, probably the largest user of vaccines against H5, followed by Egypt, Vietnam, and Indonesia. But as you can see, the disease remains endemic in those countries. I wouldn't say about Indonesia yet, I may be misquoted there, but other countries have had to deal with H5 occasionally. So the USDA is currently evaluating vaccines against highly pathogenic avian influenza virus, and they are also producing vaccines that can protect against H5. Vaccination may be useful, but it may not necessarily be sufficient to eradicate the disease if not properly implemented. A successful story with vaccination is the H7N9 vaccination of poultry in China. Essentially, since vaccination was implemented, no more human cases were detected. Okay, so vaccination does work to some extent. It may not work completely to prevent virus shedding and transmission in poultry, but in the case of the H7N9, it led to no more human cases of H7N9. Vaccines against influenza, which vaccines to use? Most of the vaccines that are being used are kill virus vaccines with some adjuvant, and there are also recombinant vectors. However, with recombinant vectors, some of the issues are pre-existing immunity against the vector itself. And the fact that we know nothing about the performance of these recombinant vectors in what is considered minor poultry, like guinea fowl, quail, geese, etc. So we know nothing about the performance of those recombinant vaccines in minor poultry that are also playing a role in perpetuation and transmission of the virus. In general, and this is again just in general, vaccines are good at preventing disease signs, but usually they do not prevent productive virus replication and spread. Another problem that most of these vaccines are very labor intensive. You cannot do mass vaccination with many of these vaccines. Therefore, we need to do better in that regard. As you know, influenza viruses keep changing. The HA mutates, drift, and is the major target of the antibody response. So most vaccines rely on this antigen, DHA, to provide immunity. And as I mentioned earlier, the recombinant vectors may be good, but they tend to be unidimensional. So they only carry an HA segment without considering that other proteins in the virus can provide both antibody-mediated and cell-mediated immunity that can add to the protection if they were incorporated into the vaccine. But since most vaccines are based on kill vaccines and the HA, one of the issues always is how is this virus mutating? Work done by colleagues at the Erasmus Medical Center, Ron Foucher, and in Cambridge, Derek Smith, they have done this very systematic antigenic characterization of influenza viruses over the years, including H5s and H3s. And they realized that those amino acids that modulate the hemagglutination inhibition activity, which is the test that typically is used to establish the antigenicity of influenza viruses, is really dominated by few amino acids near the receptor binding site of the hemagglutinin. So when we look at potential candidates for vaccines, we need to look at changes in these areas. Influenza viruses that have entered into the Americas are what they're called the 2344B clade. And this is studies that we've been doing, but essentially we are aiming to use a computer algorithm based on phylogeny to detect potential vaccine candidates that we can use. And because inactivated vaccines, in my modest opinion, are not going to be really useful and impractical if we think about potentially vaccinating wildlife, we have moved into modified live virus vaccines. In this case, what we've done is what is called genome rearrangement. So segment 2 of influenza viruses encode for PD1, and segment 7 encodes for M1 and M2 by alternative splicing. What we decided to do was to clone downstream of PD1 the M2 and incorporate a series of stop codons in the M2 open reading frame. So there is no more M2 coming from segment 7. This rearrangement itself is enough to attenuate the virus in vivo. And in addition, because our vaccine needs to have some markers in the HA that can allow us to distinguish the vaccine HA from any wild-type virus there, we put molecular markers, and these have unique sequences that do not exist in nature. There is not a single organism that has this sequence, as far as we know, that has been sequenced today. And this can act as a molecular marker, but it's also thought to reduce the fitness of the virus. So it will remain with the vaccine backbone, but it will not reassort with a wild-type virus. And in addition, we try to use immunomodulators, in this case, a cytokine that is incorporated in the segment 6 of the virus, and is thought to stimulate a better immune response. I'm not going to go into details of the experimental design because there is a lot to take in here, but essentially we prime and boost and challenge with low pathogenic H5N1 avian influenza virus. After vaccination, we see that as a control, we use a whole inactivated adjuvanted H5N1. As expected, it stimulates HI titers. The modified live virus vaccine usually is not supposed to stimulate high HI titers because most of the antibodies are expected to be mucosal type of antibodies. These are in the serum. These are supposed to stimulate better mucosal immunity. And we see that the modified live stimulates less than the whole inactivated, but the one with the interleukin tends to have decent higher response compared to the one without. And after boost, responses are boosted with the inactivated as expected and just remain more or less the same after boost with the modified live. This is also something to be expected. I mentioned before that one of the problems with inactivated vaccines is despite high neutralization titers, they not necessarily prevent shedding. And this is the case if you look at here at day 3 or day 5 post-challenge. Chickens that receive the live vaccine do have, in general, less virus, particularly the one with interleukin, than the control without vaccine or the control that received the inactivated vaccine. And by day 5, those that receive the live virus have almost no shedding, but the one with inactivated still has significant shedding, similar to the mock vaccinated birds. Another aspect that is important with these viruses is where do these viruses go? It is thought that H5N1s are going only in the respiratory tract or intestinal tract when they replicate. But what we saw is that, well, if you look at the sinuses, all 3 vaccines seem to have worked well. In the trachea, all 3 vaccines may have worked well. In the lungs, we still see some replication with inactivated group, but not with the live vaccine group. In the cloaca, we see some replication with inactivated, but not with the modified live group. But in the pancreas, with the modified live, there is almost no replication in the pancreas, but with inactivated vaccine, it seems that we have even enhanced to some extent the replication of the virus in the pancreas. So the virus replicates in many different places, and when we use whole inactivated vaccines, we may miss protection in some organs that can later lead to the virus production that may escape the initial detection. Finally, in order to try to see whether we can use this in drinking water, we have done an experiment administering the vaccine in drinking water, again through a prime and a boost. These experiments are ongoing. The vaccine is stable at a wide range of temperatures. We administer the vaccine for 1 hour. So when you look at the 1 hour time frame, either with 10 to the 6 or 10 to the 4, at almost room temperature, we see good stability of the vaccine. If we increase the temperature, some of the titers get lost, but within the 1 hour time frame, birds will be drinking enough of the virus to get vaccinated. And even at 104 degrees, there is a still significant amount of virus in the inoculum for birds to get vaccinated. The vaccine replicated on day 3. It was kind of expected that there will be some level of replication in oropharyngeal swabs, but there was almost nothing to be detected by day 5 post-vaccination. Still, that strategy for vaccination led to, this is the post-vaccination, this is post-prime, not post-boost. We don't have the data from the post-boost yet. There was enough seroconversion to say these birds really saw a virus and they seroconverted. And the same can be said about some levels of anti-MP antibodies by ELISA. So more will need to be done on this front, but I would say the poultry-adapted FLUAVS continue to be a pandemic threat. Integrated fish farming and live poultry markets contribute to the emergence of FLUAVS. So I think we need to do a better job at addressing those agricultural practices and business practices that may lead to the perpetuation of these viruses and the emergence of novel strains. The poultry-adapted H5N2 low-path viruses have a wide distribution in Asia, the Middle East, and Africa. They are respiratory flu A virus, whose gene segments can certainly increase the zoonotic potential of other subtypes. The goose Guangdong H5, a highly pathogenic virus, show a geographic range that resembles historical accounts of fowl plague in the early 1900s. So when you look through the 1900s, there have been many reports of fowl plague around the world. This is a time in which the disease was not really controlled in any way. So we're getting back to where we started, essentially, in terms of what these viruses are able to do. They also show an unprecedented host range, but I would say that risk to humans remain low. I think that infection in mammals, although sometimes lethal, are likely due to inoculation with high virus dose. And I think the same argument can be made for the detection of or death in raptors and scavenger bear species. But these viruses do have or may have big impact on wild bird biodiversity. Vaccination of poultry per se remains controversial, at least in some countries. But we think that modified live virus vaccines, if they can be applied through mass vaccination, can offer an opportunity to better control the disease where kill vaccines or other vaccines are not doing the job. Sorry if I took too long. I would like to thank people in my lab, past and present, that helps me with the work that we do. And thank you. I don't know if you guys have any questions.